The brain is one of our most heavily guarded organs. Yet, just by aging, a process everyone is doing at the moment, we lose neuronal connections. One reason why we don't see 130-year-old people is because even our best medical tools and findings have trouble maintaining brain health and function over an extended lifespan. For postmenopausal women, the risk for neural breakdown is especially high. Estrogen deficiency increases brain insulin resistance and neuroinflammation. It increases pro-inflammatory cytokines, TNF-alpha, IL-1 beta, and macrophage inflammatory protein 1. So, what can we do about it? This paper, Intermittent Fasting Protects Against the Deterioration of Cognitive Function, Energy Metabolism, and Dyslipidemia in Alzheimer's Disease-Induced Estrogen-Deficient Rats, cuts to the chase by considering fats. Recall that there are two types of white adipose tissue, subcutaneous and visceral, which are respectively beneficial and disruptive to metabolic activity. Existing literature concludes that estrogen loss coincides with 1. More visceral fat relative to subcutaneous, 2. Weight gain that can be suppressed with hormone replacement therapy. Hormone replacement therapy has deleterious effects, however, which prompted the researchers to evaluate the effectiveness of intermittent fasting. To fast intermittently, a person refrains from eating 1 or 2 days per week. In that sense, it is less of a diet than it is an eating pattern. The researchers hypothesized that intermittent fasting would have an effect on metabolic disturbance in memory. They chose female rats as their study model and randomly assigned the rats to Alzheimer's and non-Alzheimer's beta amyloid. In nerve cells, beta amyloid is a transmembrane component of a larger precursor protein. It aggregates into plaques found in Alzheimer's patients. The researchers proceeded with a series of tests and analyses.
For the experimental design, Sprague Dally female rats aged 10 weeks were used and given a 1-week acclimation period in the steel cages. The animals consumed water ad libitum, which is a diet that's available at all times, and then they were assigned to their experimental diets, which took place over 8 weeks. The high-fat diet had 38 energy percent or EN% carbohydrates, 16 EN% protein, and 48 EN% fats. At 11 weeks old, the rats had an oviaryectomy, which is a removal of both ovaries, and these rats were termed the OVX rats. The OVX rats were then divided into 2 groups of the high-fat diet. Ad libitum, which was free to eat whenever, or intermittent fasting, a restricted diet over a restricted amount of time. 2 weeks after the assigned feeding groups were introduced, rats of one group had an ICV infusion with beta amyloid 2535, which produces Alzheimer's disease, or beta amyloid 3525, which does not produce Alzheimer's disease, and these rats have normal cognitive function. An ICV injection is an invasive injection technique of substances that are directly into the cerebral spinal fluid and the cerebral ventricles in order to bypass the blood-brain barrier, which was done in this study with beta amyloid. The ICV infusion was done when the rats were anesthetized using a stainless steel cannula, which is a thin tube inserted into the vein or body cavity to administer medicine, and a stereotactic device, which uses a set of coordinates that allows for the precise location of brain sections, where the beta amyloid was implanted in the CA1 subregion of the brain, which is the region in the hippocampus where CA1 neurons are found, and these neurons are involved in the formation, consolidation, and retrieval of hippocampal-dependent memories. The diets were provided for 4 weeks after the ICV infusion of beta amyloid, and the study therefore had 4 groups which are shown. The first one was beta amyloid 2535 plus the ad libitum diet, beta amyloid 2535 plus the intermittent fasting diet, beta amyloid 3525 plus the ad libitum diet, and then beta amyloid 3525 plus the intermittent fasting diet. The researchers chose to use a time-restricted intermittent fasting model, which was restricted to a 3-hour time period. In the IMF groups, rats had a high-fat diet for 3 hours from 7 to 10 p.m., and then the food was removed until 7 p.m. the following day. Gene expression in the hippocampus. The hippocampus was collected from 5 rats after injection, and specific genes associated with inflammation and degradation of the hippocampus were amplified using a mixture of cDNA from each rat's hippocampus, primer for TNF-alpha, IL-1 beta, and IL-6, which are all markers of inflammation, and cybergremix using PCR. For the histopathological analysis of the hippocampus, at the end of the experiments, the brains of the rats were given 4% paraformaldehyde and left in a 30% sucrose solution. The beta amyloid deposition in the hippocampus was detected with anti-beta amyloid antibodies using immunohistochemistry. Beta amyloid accumulation is shown as the percent beta amyloid positive cells in the hippocampus. For the immunoblot analysis, hippocampi were collected, lysed, and protein was collected, and electrophoresis was conducted, and then samples were reacted with multiple antibodies. Intensity of protein expression was measured using ImageQuant TL, which is an image analysis software for gels, blots, arrays, and colony counting. Statistical analysis was also performed, and SAS software was used as well as ANOVA. One way ANOVA was used in the study was to assess the metabolic effects of intermittent fasting and beta amyloid infusion into the hippocampus in OVX rats at a single time point.
Here we're looking at results for beta amyloid deposition in the hippocampus. Beta amyloid deposition is a biomarker for Alzheimer's disease. We can see that in the 4 groups of mice that were all infused with beta amyloid, immunoreactivity of beta amyloid was detected mainly in the 280 models of mice. Comparing the 2 groups of 80 mice, by having an IMF diet, the amount of plaque was significantly lower compared to normal diet. However, the diet did not make a difference in non-80 mice models.
At the 3rd week after beta amyloid infusion, rats were assessed for short-term memory using passive avoidance apparatus, which is a 2-compartment dark light shuttle box. Short-term memory was measured by the latency time to enter the dark chamber at the 2nd trial when an electric foot shock was not delivered, and spatial memory was evaluated using a Morris water maze test. In this case, a shorter latency time and longer staying time indicates better long-term memory and long-term spatial memory. Bone mineral density measurement. Using a DEXA scan, bone mineral density in the right femur and knee, fat mass in the abdomen, hip, and leg, and lean body mass were measured in the anesthetized rat. This was done before the ovarectomy and after 6 weeks of treatment. Locomotive activity. 3 days after the water maze test, locomotive activity was tested using an MH1053 activity monitor, which measures activity by breaking 3D arrays of infrared beams in a cage. The rat acclimatized to the cage for 30 minutes, and the activity was then tracked for 1 hour.
The researchers evaluated the cognitive function of these mice. On the left panel, we can see results for the avoidance test. Alzheimer's disease mice took longer to enter the dark room in the test compared to non-AD mice. However, with the IMF diet, it partially prevented the increase in time in the 1st and 2nd trial, indicating that Alzheimer's disease decreases short-term memory and IMF may help to prevent this functional loss. Now let's look at the right panel. Here, it shows us delayed latency time to locate the platform. We can see that 80 mice took significantly longer time in general compared to non-AD. Similarly, having an IMF diet prevented the delay. In addition, they also observed that the duration of time mice stayed in zone 5 was longer in non-AD mice than Alzheimer's disease, as well as 80 IMF stayed longer than 80 normal diet groups. These results show us that plaque formation decreases spatial memory. Researchers looked at bone mineral density, lean body mass, and fat mass in different body areas. On the left panel, we can see changing bone mineral density presented on the y-axis. After ovary removal, bone density was lowered in both the spine and the femur. When comparing IMF diet to normal diet in both groups of AD and non-AD mice, IMF diet tends to decrease bone mineral density. This change was significant in the left femur indicated by the red arrow. Middle panel showing us changing lean body mass. During the experiment, all mice had decrease in lean body mass in the hip area, whereas increase in the left leg. IMF diet had significantly more lean body mass compared to normal diet in both AD and non-AD mice group. Changes in fat mass were looked at 2 different areas, the abdomen and left leg. While in the abdomen, IMF diet mice had significantly less gain fat mass throughout the experiment, in the left leg, there was no difference observed between the 4 experimental groups.
I will now lead the discussion of the paper. As mentioned previously, estrogen deficiency can cause beta amyloid accumulation. From this flowchart, we can see that beta amyloid accumulation is a center of all metabolic and cognitive problems. It can directly and indirectly cause the symptoms observed in Alzheimer's, as you can see in the blue circles. Based on the result of the study, we know that intermittent fasting can provide some relief by decreasing beta amyloid deposition, which in turn improves memory function and other cognitive functions. In general, intermittent fasting diet groups achieve a more desirable body fat composition, with more lean body mass and less visceral fat in the abdomen. However, this diet can worsen cholesterol-related problems. In Alzheimer's, LDL cholesterol and triglyceride levels are higher than non-Alzheimer groups. As shown in the red box below, with the fasting diet, triglyceride levels decrease along with the healthy cholesterol HDL, but the LDL level remains similar with the no fasting group. There's also a decrease in bone mass density associated with the intermittent fasting. The mechanism behind this decrease is still unknown. It could be triggered by the loss of estrogen and further escalated by the fasting process. Moving on to the critical analysis of the paper. This is the first study in the field to examine the effects of intermittent fasting on estrogen deficiency symptoms. They used randomized controlled trials, which can increase the quality of their research. and they were able to emulate the symptoms of Alzheimer's in the rat models. I think this is a strong point because they were able to recreate the problems in their patients in the animal models, which further adds to the quality of research. However, I also find many flaws in their methodology. A few obvious flaws are the small sample size and the short duration of the study. Moreover, they used young female rats in the study, where the target populations are postmenopausal women. They also did not do any assessment after the beta amyloid injection to make sure that the rats have Alzheimer before continuing the experiment. And lastly, this is the mistake that I find to be the most crucial. They did not measure the amount of beta amyloid right after the injection. They only measure beta amyloid after the diet ended, since the brain is a very guarded area and it actively filters out impurities. The amount of beta amyloid that they put in the brain may not all end up in the hippocampus. They should use markers to locate the position of amyloid plaques in the brain. Depending on where it accumulated, it can affect the progress of the disease differently.
To summarize, the authors' conclusions from this study surround the existence of 80 impaired cognitive and metabolic decline and dyslipidemia in estrogen-deficient rat models, of which intermittent fasting acted protectively to counter the negative effects of deterioration. While estrogen deficiencies in menopausal women correlates with visceral fat accumulation and insulin resistance, by upregulating hippocampal insulin signals and thereby inhibiting beta amyloid plaque formations, IMF was able to improve memory functions, as well as other concerns regarding Alzheimer's disease. Unfortunately, there is a drawback that came from IMF in relation to cholesterol. The IMF diet decreased the amounts of beneficial HDL, whereas the harmful LDL levels were less affected by this diet. One limitation of this experiment is that, although intermittent fasting protected against cognitive decline and insufficient energy metabolism, further studies are needed to elucidate on the effects of intermittent fasting in menopausal women with disruptions in metabolizing glucose, such as in glucose-intolerant or diabetic women. Due to the previously stated concerns in cholesterol management with intermittent fasting, this diet may cause harm to women with cholesterol and metabolic problems. In addition, another clear limitation in applying this research onto a human population of women is that this study was conducted on rodent models rather than people. Therefore, more research should be done in order to clarify the effects that IMF would have on target patients. In comparison, however, to hormone replacement methods that the authors had mentioned earlier, this study focuses on IMF diet treatments to alleviate the problems that arise with estrogen loss, since hormone replacement therapy is known to have even more harmful side effects as a treatment for weight gain. One commentary on studies that do examine hormone replacement therapy in response to estrogen loss shows comparable results to this study. As opposed to the intermittent fasting diets, in which the individual refrains from eating for a select number of days per week, hormone replacement therapy involves treatment with estrogen and progesterone to combat AD-related decline. One study showed estrogen's role in neuroprotection through clinical studies, in which a cohort was assessed for the beneficial effects in women taking estrogen, leading to a reduction in dementia. However, another study showed that estrogen correlated with a slight increase in impaired cognitive abilities onto a different cohort, with no significant effect on dementia. The authors concluded from these findings that the timing of taking estrogen could explain the discrepancies between different populations of women using estrogen for neuroprotection. Therefore, future IMF studies are crucial for heightening cognitive function in AD, and conduction of more IMF research is important for the creation of alternative treatment methods of riskier options, such as hormone replacement therapy, in Alzheimer's disease health due to estrogen loss.